No, sure. No. Come on, stop it. Don't be grumpy. Oh, hello. Oh, yeah. Give a treat. Russia, don't we stop?
Wanneer ek het vroeg geleer wanneer hulle my kan sien.
Recording in progress. Okay, but I'll keep watching this one, but I'm just scared now to change anything because, yeah. If it was a normal plain one, then it's easy. But now it's a little bit complicated. Okay, sister.
Die lekker plek in die hande van 2,7 kilo.
Okay, I think we can start. Welcome everybody to the third educational session of the Blood Transfusion Genomic Consortium. It's our third educational session, and the topic will be the iron homeostasis in health, and will be hosted by Colin and by me. And today we have 4 speakers. The first 2 speakers will talk about the iron, and the last 2 speakers will show some results of our array. The first talk will be by Dorine Swinkels. She is a professor in the experimental clinical chemistry at the Radboud University Medical Center in Nijmegen. And she's head of the Iron Expertise Center of Sanquin in Amsterdam in the Netherlands. And the second speaker will be Elias Allara. He's a clinical research associate at the Department of Public Health and Primary Care in Cambridge in the United Kingdom. And after this, we will have I can't see my second slide. I don't know why. Yeah. After this 2 talks, there will be a possibility to ask questions. You can do that either verbally or in the chat box, preferably in the chat box, because then the presenters can read it and probably give a better answer. And this is followed by a co-presentation of William Lane. He's an assistant professor at Harvard Medical School at Brigham Women's Hospital in Boston in the United States, and Nicholas Gleadall, who's an assistant professor in clinical genomics of transfusion and transplantation at the Department of Haematology at the University of Cambridge in the United Kingdom. The Blood Transfusion Genomics Consortium is an international partnership between blood services, research institutions, and industry leaders. And it's our aim is to improve the safety and efficiency of blood and platelet transfusion by introducing large-scale comprehensive genotyping array for donor typing. And this is what you will hear about in the next 4 talks. So I will give the floor to Dorine. And now I should stop presenting. Can you? Yeah. Okay. Thanks.
Okay. Good day to you all. I will share with you some knowledge on iron in health and disease. So as you all might know, iron is essential, but at the same time, it's also toxic. It's crucial for life, and it has various roles. So if I ask my first year students, they all say it's involved in hemoglobin, but it does far more. It's involved, it's essential even in DNA synthesis, respiration, many metabolic processes, and also, for example, in heme, the synthesis of iron cluster, iron sulfur clusters, and also involved in functioning of very many enzymes. So it's essential, it's toxic, because it leads to free radical damage. So iron levels must be tightly regulated, both at the cellular and systemic level. So normally I don't do this, but for this presentation, I'll present you my content. So I will go into iron homeostasis. That will be the first part. And secondly, I will also talk a bit about biomarkers that can be used for the readout of iron homeostasis. So let's zoom in and start to zoom in. Let me see how I get this out. Okay, so we start with systemic iron homeostasis, and that involves iron absorption in the gut and iron recycling. So if you go to this slide, in the middle, we have iron bound to transferrin. And this iron in the circulation is lost. I mean, 1 to 2 milligrams a day we lose, and that's more in premenopausal women, of course. This iron is used for by all the tissues in the body, but mostly by the bone marrow to produce red blood cells. And when they become senescent, the red blood cells will be taken up by the reticular endothelial system, the macrophages, and iron will be released in the macrophages and recycled into the circulation. Because we lose iron every day, iron is replenished by iron uptake in the gut. If we have sufficient iron, iron will be stored in many tissues, in the macrophages, but also in the hepatocytes, in the liver. So until 20 years ago, we didn't know whether, we suspected it, but we didn't know whether iron uptake from the intestine and iron release from the macrophages was regulated. But then in around 2000, 2001, hepcidin was discovered. Hepcidin is a small peptide hormone that's produced by the hepatocytes, and it's regulated by various stimuli. I will go into that in a few slides later. And what hepcidin does, it blocks iron uptake from the intestine, and it blocks iron release from macrophages. So when hepcidin is high, iron in the circulation will be low, and iron will be sequestered in the macrophages. If iron is low, hepcidin iron uptake will be high, and there will be much iron released from the macrophages, so there will be high iron in the circulation, and this will lead to iron overload. So high hepcidin, low iron, low hepcidin, high hepcidin, low iron, low hepcidin, high iron. That's important to remember. Let me see how I go to the next slide. I can't. Oh, here, sorry. So this is a bit of a complicated slide, but what we see is the hepatocyte. This is the membrane, and this is the nucleus. And the nucleus produces hepcidin, and it's controlled, its production, by various processes that involves many proteins either on the cell membrane, but also in the cell. And these processes are mainly controlled, as you might suspect, by erythropoiesis, by iron status of the body, and by inflammatory processes. So, for example, if the erythropoiesis is high, there's a high need of iron, the bone marrow will produce ARFI, and ARFI will then interact with a BMP SMAD pathway and lower hepcidin levels. If, for example, there is high iron status, hepcidin will be increased to make sure that there will be less iron uptake from the intestine and less release from the macrophages. So all these processes are involved in the regulation of this important iron regulatory peptide, hepcidin. So if we have a defect, for example, in HIV, there will be less hepcidin production and there will be iron overload, hemochromatosis. When we have a defect in matriptase 2, that is encoded by the gene TMPA6, hepcidin will be high and we will get an genetic form of anemia, iron refractory iron deficiency anemia. If I then go to the next slide, we can see the systemic iron homeostasis again. We see iron release from the enterocytes. We see iron recycling by the macrophages. And we also see that the hepatocytes that stores iron when there's sufficient iron and releases iron when iron is needed in the circulation. And as you can see in this slide, many cell types are involved in systemic iron homeostasis. And each of these cell types have their own cellular iron uptake mechanisms, iron storage mechanisms, and iron release mechanisms. I will go into that in the next few slides. But first, before I do that, I will explain to you that cells have their own iron regulation. It's a very sophisticated system, and it involves both iron responsive proteins and iron responsive elements. So in the absence of iron, of low iron, the IRP proteins of the iron response proteins are present. And what they do is they bind either to the 5 UTR IRE sites of various genes, or to the 3 extent UTR IRE of other proteins. So, for example, if there's low iron, of course, the ferritin, that is an iron storage protein, will be decreased. So it binds, the IRP binds to the IRE of ferritin, and therefore, and this binds the inhibits the translation of ferritin. It also binds, for example, in the presence of low iron in circulation to transferrin 1, and this inhibits the decay of the transferrin receptor 1, and it's very logical because transferrin 1 is involved in iron uptake from the circulation. And of course, if you have low iron levels, you want to increase the iron uptake by the cells. So it is a very sophisticated system that is designed to regulate the intracellular iron homeostasis system. If we then focus on cellular iron metabolism of the gut, in this case of the enterocyte, what you can see is this is the apical site of the enterocyte, and this is the bloodstream. And at the apical site, of course, there is our food entering, passing by the apical site of the enterocyte. And the iron that is in the 3+ status is then reduced to iron 2+ and taken up by the devolent metal transporter 1. This iron is going into the cell, probably chaperoned by poly-RC binding proteins, and then either stored in a storage protein, ferritin, or transported to ferritin, that is an exporter protein, and to the circulation. Also, the hypoxia-inducible factor 2 is involved, and when it's present in case of low iron or low oxygen, there will be more transcription of DMT1 and more transcription of ferroportin, so more iron will enter the body. If we then go to the iron recycling macrophages, iron is mainly entering the cell by recycling by the uptake of the engulfing of senescent erythrocytes. Then heme is coming into the phagolysosome and transported into the cytoplasma by the HRG1. This heme, heme is then coming into the cytoplasma, and heme is then degraded by hemoxygenase 1, and the iron that is released is either again stored in ferritin in the macrophages or released by the macrophages into the circulation. And for this purpose, iron 3 is reduced to iron 2+ to be released by the ferroportin. So also the macrophages have their own system of iron recycling that involves many proteins. If you then go to the hepatocytes, iron is taken up either by the transferrin receptor that binds iron-bound to transferrin in the circulation, or if there is transferrin is saturated, iron will be freely circulating in the blood, and that will be taken up by the CYP14 transporter. Both forms of iron are again stored in ferritin in the hepatocytes or transported to ferroportin and released by the hepatocytes. And of course, the hepatocyte is also important for the production or synthesis of hepcidin, as I explained to you in the previous slides. So if we then focus on iron metabolism in erythroid precursor cells, what we see is that iron is taken up from transferrin iron in the circulation. That is endocytosed, and by acidification of the in the endosome, iron is released into the cytoplasma by DMT1, and transferrin is recirculated to the plasma. Now, iron can also enter by pinocytosis of ferritin. That's mostly observed in vitro. And iron that is in the cell can be either stored again in ferritin, exported to ferritin, and exported by ferroportin. But what's most important is that iron is utilized in the mitochondria of the erythroid precursors. In the mitochondria, it's entering the mitochondria by metaphoron 1, that is stabilized by ABC by B10. And iron in the mitochondria is either used for the production synthesis of iron sulfur clusters that are essential for many metabolic processes, both in the mitochondria and in the cytoplasma. And iron is also used for the production of heme. That involves many precursors, and then the heme is transported to the cytoplasma and then bound to globin to form hemoglobin. And of course, the hemo and the sulfur clusters are also exported to the cytoplasma, used in the for the energy metabolism of the mitochondria, but also exported to the cytoplasma to be used for client proteins. What is important also for the young erythroid precursor cells is that they also have transferrin 2 receptor on the membrane, and this also senses the iron saturation of transferrin. And when it's low, then the production of RBCs, of red blood cells, will be decreased, to be sure that not all the iron in the body is used for erythropoiesis. Moreover, when there is low iron in the circulation, ERFI will be produced, erythroferrone, and erythroferrone interacts with the upstream pathway of hepcidin, and as a result, hepcidin will decrease and more iron will become available to replenish the iron in the circulation. A complicated system. So in summary, enterocytes, macrophages, hepatocytes, and erythroid precursors all have the private proteins involved in iron uptake, transport, regulation, and storage. But they also share certain proteins. For example, ferroportin, ferritin, DMT1, and the transferrin receptor 1. And the mitochondria has also specific proteins involved in iron regulation. And if these proteins are are aberrant or not functioning anymore, it will result in either anemia, iron overload, the combination of anemia and iron overload, neurological diseases, and also other metabolic disorders. So this was my part about, let's say, iron metabolism in a nutshell. And now I will just have a few slides on biomarkers of iron metabolism. So as you all might know, we measure ferritin in the circulation to get some insights in the amount of iron that's stored in the body. If we then go to the circulation, iron in the body, in the circulation, is bound to transferrin. And the extent to which transferrin is saturated with iron is measured by the transferrin saturation, or so-called TSAT. And when TSAT is fully saturated, non-transferrin bound iron, or its oxidative active protein labeled plasma iron, will be formed. Toxic iron species. And what we can also measure is, we can also get an impression of iron available for the bone marrow by measuring soluble transferrin receptor, or a precursor of heme, that when there's not a sufficient iron will not acquire iron, but instead of iron, zinc. So the zinc protoporphyrin amount is a marker of iron deficiency. And of course, we can also measure erythropoietic markers like HB, MCV, and MCH. So all these markers can give us insights in the iron status of the body. And of course, we can also measure hepcidin. As you know, hepcidin blocks ferroportin, especially in the stores, macrophages, and the enterocytes. But when there is insufficient iron or hypoxia, erythropoietin will be produced in the bone marrow by the red blood cells that will block hepcidin, and as a result, more iron will be released by the stores, and more iron will be taken up by the gut. So hepcidin we can measure, and hopefully, and some labs can also measure erythropoietin, but this in general, to get more reliable assays, more work for erythropoietin, more work has to be done. There's one last point I want to make, is that many of the markers we use to measure, to get insights in iron metabolism, are not standardized nor harmonized. So this indicates that if we have a certain sample and we send it to several labs, we get different results. Harmonization will then result in similar results for the different labs. Or if we can even, standardize by using adequate calibrators, we get the true value. And if we then go to the next slide, you can see in this slide that various markers are not very well harmonized, nor very well standardized. So it's important to take that into account. And even ferritin can give different results in different laboratories. It's not optimally standardized nor harmonized. There are calibrators. So it's possible, but it's not fully implemented. And the same is also true for hepcidin. So take that into account when you have your biomarkers measured in a certain lab. It's difficult to compare between laboratories. Then I go to my last slide, to the summary. So iron is regulated at both the systemic and cellular level. It has links to oxygen sensing, and this all to ensure sufficient iron supplies for erythropoiesis, mitochondrial function, and more. Various cells cells share similar mechanisms and related proteins for ion uptake, storage, and export, but many cells, specific pathways and proteins exist. Defects in ion regulatory pathways lead to anemia, ion overload, and various other disorders. And various ion biomarkers and DNA analysis, of course, also can provide insight in the cause of ion disorders. But it is important to note that for some biomarkers, measurement results may differ between laboratories. Thank you for your attention. I'm happy to take questions after the whole session.
Thank you very much, Dorine. So we will move to our next speaker, and that will be Elias Allara from the Department of Public Health and Primary Care at the University of Cambridge, who will talk to us on genome-wide association studies of iron hemostasis biomarkers.
Thank you very much for having me. I'm going to quickly share my slide. Hopefully you're able to see them.
Yes, we can see that.
Yes, we can.
Okay, fantastic. Thank you very much for having me. I'm a clinical epidemiologist, the cardiovascular epidemiology unit at the University of Cambridge. I'm going to tell you, to give you some information about genome-wide association studies of iron homeostasis biomarkers. The first study I'd like to talk about is one study was published in 2014, and it was the first large-scale genome-wide association study at the time that was focusing on common ion trait, specifically ion transferrin saturation, and ferritin. The study was conducted in over 50,000 participants. The authors found 11 loci associated with iron trait. Some were known, others were new, 5 of them. Some loci, interestingly, were available in all phenotypes. For example, HFE is number 4 in the Manhattan plot here, and TMPR SS6 number 11 in the plot here, likely reflecting the key role in iron homeostasis. Other loci instead were more phenotype specific, such as SLC40A1, which is number 1 in the plot here, which codes for ferroportin and was found to be associated with ferritin and transferrin. The authors also found overlap with other phenotypes, such as red blood cells and lipids. And we are going to see in the next slide why this can be of clinical relevance. More recently, there's been one large-scale study in over 240,000 participants, still focusing on common ion traits, the same as in the previous slide. This study found association in 56 loci, of which 46 are new. And they also found overlap of loci across phenotypes. In addition to HFE and TMPR SS6, also dual X variants were found to be associated with all traits. And dual X2 is quite an interesting locus. It's thought to be involved in interaction between the gut microbiome and the intestinal mucosa, and therefore influencing iron absorption. The latest of this series of study was published earlier this year in approximately 260,000 participants, and found association with over 100 loci, of which 19 with new variants. I wish we had a little bit more time to talk a little bit more in detail with this, but I think the core message from this few slides is that in in the past years, we have increased our knowledge of the genetic architecture of a common ion trait with increasingly larger sample sizes and increasingly larger number of discovered genetic loci. In terms of other ion traits, such as more emerging, such as soluble transferrin receptor, we are not there yet. The most recent study I was able to find was published in 2011 and found association with HFE and TMPRSS6, the usual. And in addition, the strongest association was found in PCSK7, so it's harboring the strongest association found in the GWAS, which is it is quite an interesting locus involved in the shedding of transferrin receptor and generation of SDFI. In terms of hepcidin, that Professor Swinkels has abundantly emphasized its key relevance in iron homeostasis, and it would be useful to know more about the genetic architecture. Unfortunately, we are not there yet. The most recent study was published in 2016. Comprised over 6,000 participants, and this was a quite comprehensive study. It's up to 38 million SNPs were analyzed, and unfortunately, there was no common or rare exonic genetic variants that were able to explain a large proportion of phenotypic variation in hepcidin. And the author mentioned that this may be likely related to the fact that hepcidin variability is most likely caused by variation in non-genetic factors and rather than genetic factors. And they also encourage researchers to provide more evidence to improve our understanding of the genetic makeup of this important biomarker. So what we can do with this large set of genetic variants, and not only improve our understanding of ion biology and therefore possibly provide some targets for therapeutic opportunities, but also we can use this variant to generate polygenic score or PGS that enable estimation of individual genetic liability for specific phenotypic trait, in our case, ion trait. The process is possibly more straightforward than you may think, and it's in its core. The core process can be summarized in 2 steps. The first, we select the variants associated with the exposure of interest, in our case, ion trait. We take the top association, and we have a table like this one. I hope you can see my mouse cursor. In the first column, we have the genetic position of the variant. In the second, the effector allele. In the third, the effect size. The effect size represents the change in the phenotypic trait for each 1 unit increase in the effector allele. Because we have 2 copies of each chromosome, we may have either no effector allele at all, or 1 copy of the effector allele, or 2 copies of the effector allele. So either 0, 1, or 2. Secondly, for each individual in the data set, we multiply the product of the genotype, either 0, 1, or 2, with the estimated effect size, the third column here, for each genetic variant included in the PGS, and then we sum this number. The result is the individual genetic liability for a specific phenotype. This is one example using only 2 variants from the above table and 2 participants. This focus on participant A. For the first SNP, this participant has the genotype AG, which means that she has 1 effector allele and 1 non-effector allele. And for the second SNP, this person has CC, which means that she has 2 effector alleles. So we want to calculate the polygenic score for this very simple PGS. Then we multiply the number of alleles for the first SNP with the effect size of the first SNP, and we have a 1 times 0.031. And then we sum this with the multiplication of the number of effector allele for the second SNP, which is 2, times the effector allele for the second SNP, which is 0.037. We sum all of this, and we have the polygeny score for this person, which is 0.105. Obviously, we have many more individuals and many more variants, just to give you an idea of how polygeny score is calculated. What can we do with the PGS in biomedical research? I could think of at least two possible applications. First is expedition certification, and the second is etiological research. I admit I'm not an expert of the first type, or the first application. There are many excellent colleagues who know a lot more than me, but hopefully be able to give you a short introduction. So essentially, we divided participants into equal parts, such as quintiles based on the PGS. And so we have this distribution, like in the left-hand side figure here, and we divide the population in equal parts. In this case, in 5 parts, but can be also 10 parts, depending on what we decide. And then we predict the risk in each quintile, and we can have a figure similar to the right-hand figure here. And as you can see, the participant with the lowest quintile of PGS also have on average a lower risk of the trait of interest. In our case, is hypertension, but can be whatever. The second application is in etiological research, and using PGS for this is one possible method to estimate causal association in a Mendelian mutation framework. I'm going to tell you more about this in the next couple of slides. So how we do this, we regress the outcome of interest. Can be any trait that we think may be a biomedical consequence of iron. And we regress the outcome on the PGS using a linear equation similar to this one. In this case, the beta-1 coefficient represents the unbiased genetically predicted effect of the exposure of the iron trait on the outcome of interest. In this case, the PGS usually based on much fewer variants than for risk prediction, because we are interested in causality. So ideally, we want these variants to be strongly associated with a trait of interest, or even possibly have a plausible role in the biology of the trait. And secondly, this method requires a data set with individual participant data, because we need both. Like you can see from this equation, we need data for both the outcome and the genetics. Let's have a step back and talk a little about Mendelian mutation, how it can be used to assess the potential causal effects of iron trait. This slide presents a comparison with randomized trials. Of course, the two methods are drastically different in terms of study design. So a randomized trial is experimental in nature, while Mendelian mutation is observational in nature. So there are some striking differences. But I think conceptually, the two designs can be compared to gain a better understanding of how Mendelian mutation works. So in a randomized trial, we randomize people into groups, like control or treatment groups, usually, in the simplest form. In the control group, we expect people to have higher levels of the risk factor, for example, blood cholesterol, if our treatment of interest is lipid lowering drug. And consequently, we expect higher level of the outcome of interest, for example, cardiovascular disease. In the treatment group, we expect, of course, lower risk of the risk factor and lower risk of the outcome. In Mendelian mutation, in its simplest form, essentially we have randomization by genetic variant. This is one single variant. Of course, can be more than one. Like we can replace this with a polygeny score, but let's focus on the simplest example. So assuming the genetic variant is beneficial, when the variant allele is absent, then we have higher level of the risk factor, and therefore a higher level of the outcome. And the opposite happens when the variant allele is present. How can we apply MR in practice? So the process is, the first part of the process is very similar to the one that we use to generate PGS. So we select the variants associated with exposure of interest, but there is one important difference here. We are interested in the variants that are strongly associated with exposure of interest. This is because we are interested in causality rather.
I think that's better. That's better.
Sorry, can you hear me?
We can hear you now, Elias. Thanks.
Yeah, sorry. For each of these variants that we select that are strongly associated with iron trait in our example, we get the effect on the exposure, that is third column here, and the effect on the outcome. It's our outcome of interest, for example, cardiovascular disease or diabetes, whatever. And imagine we have a fourth column here with the effect size with the outcome. And then we estimate the genetic effect on the outcome per each 1 unit increase in genetic effect on the exposure. Apologies, it's a bit of a mouthful, but essentially what we do is we have a plot like this in which we have genetic effect on the exposure on the x-axis and genetic effect on the outcome on the y-axis. And then we try to obtain the best fit between all these values. And this will be the red line plotted here. And we are interested in the slope or the red line, which is our parameter of interest in Mendelian annotation. The interesting thing of MR is that it's two-sample MR, this method does not require individual participant data. So this facilitates the sharing of data and enables accounting for a heterogeneity between variants. So in this plot, I only showing the point estimate, but in reality, we also have measures of precision and confidence interval. Then it's possible to account for heterogeneity between variants as well using this method. Mendelian randomization, like any statistical methods, need to satisfy some assumption. The first one, it's already been mentioned. So the genetic variant needs to be strongly associated with the risk factor. If this does not happen, then we may have an issue that we call weak instrument bias. It may invalidate this assumption. The second assumption consists in genetic variant not being associated with confounders of the risk factor outcome association. This is quite important because if the genetic variant is associated with confounders, then for each, we observe a spurious increase in the risk factor and simultaneously a spurious increase in the outcome. And we think that the risk factor may cause the outcome, and in reality is not, because we have an association with a confounder. The third assumption consists in the genetic variant not being associated with the outcome. And similarly, if we have a genetic variant associated with the outcome and the same genetic variant is also associated with the risk factor, then we may observe an increase in the risk factor, an increase in the outcome that is not driven by the association between the risk factor and the outcome. It's not driven by this, but it's driven by the dotted line here. So we do not want the genetic variant to be associated with confounders or directly with the outcome. The genetic variant ideally should be associated with the outcome only via the risk factor of interest. And one problem of genetic studies is that sometimes we have pleiotropy, that is, we have more than one genetic effect on the same variant. And this may invalidate the second and third assumption of Mendelian randomization. In terms of MR studies of iron-related trait, in the past few years, there have been several interesting investigations that overall have found that higher genetically predicted iron levels have been associated with lower risk of coronary heart disease, red blood cell-related trait, hypercholesterolemia. I can see the forest plot here, with lower risk of anemia, lower risk of hypercortisolemia. But also higher risk of stroke and infections. I can see the forest plot of infection here. However, the studies unfortunately were unable to involve several biological pathways and could not be considered because of lack of updated genome-wide association studies. The largest studies were published in the past couple of years, and this is probably why we don't have yet updated Mendelian methylation studies. And most studies used only 3 genetic instruments from 2 loci, HFE and TMPRSS6, on the ground that they were the only that were associated with all the common iron traits from previous GWAS. And finally, the previous studies unfortunately were unable to characterize pleiotropic effect. So importantly, the same approach that I've discussed can be applied also to clinical phenotype. So, for example, restless leg syndrome, a paper from 2017 found 13 new loci associated with RLS, and they found also genetic correlation with several other traits, including positive correlation with several neurological and psychiatric diseases, and an inverse genetic correlation with years of schooling and other education-related variables. What's next? The moment in our group we are working to improve our understanding of the genetic architecture of emerging iron traits, like hepcidin and STFR, and we're also working to investigate the role of iron-related biological pathways and systemic iron status on human health. In summary, genome-wide association studies are powerful tools to identify the determinants of iron regulation and contribute to understanding the etiology of iron disorders. Polygenic scores and two-sample Mendelian normalization approaches can be useful to understand the consequences of iron on disease, although weak instrument bias and pleiotropy need to be properly addressed. The same approaches can be applied also to clinical phenotypes such as RLS. And finally, ongoing efforts are expected to improve our understanding of emerging iron traits and the biomedical consequences of iron. Finally, I'd like to acknowledge the contribution of the people contributing to this research, including colleagues from the University of Cambridge, from Radboud UMC, Imperial College London, University of South Australia, as well as all the study leaders and participants. Thank you very much.
Thank you very much, Elias. Now, we'll open the floor for questions. And what I can say is that if you have any questions, if you don't want to say them out, you can put them in the chat. I can see one in the chat from Connie. It was to Dorine. Do you have any opinion on testing blood donors for genetically associated iron traits?
Yeah, so I think I already
I've seen that
in the chat. Maybe I can add a bit to that. Of course, blood donation is not extremely physiological. So that means that if you can blood donation really makes in the end everybody iron deficient, especially if you do it frequently, of course. But of course, there are donors that can donate more frequently than others, and there's obvious factors like iron intake, especially meat, and premenopausal women, of course, can also have less donations. But there's also genetic factors, and there's more likely genes like HFE and TMPRSS6, of course, as I mentioned in my talk, but there's also more rare variants that probably can contribute to susceptibility to iron deficiency. So I really think that, let's say in the end, genotyping donors may help to personalize donation.
Thank you, Dorine. There's a question from Willem. It was interesting to note that the IL-6 receptor is an association signal for iron markers. Is this through inflammation and IL-6 levels and its feedback on hepcidin?
Yeah, I'm not an inflammation expert, although inflammation interacts, as you all might know, very intensively with iron metabolism. And yes, the IL-6 receptors is involved in the sensing of inflammation by the hepatocytes. And yeah, IL-6 is involved in, has a relation with iron markers because of hepcidin. That's an important, as an important factor. There might also be some local effects in the macrophages, of course. But I think the hepcidin is the strongest, yeah.
Thank you. And we have a question from Sarah Trumpeter. From a clinical perspective, we see a lot of people with low or borderline low ferritin. That has a very symptomatic and iron treatment improves symptoms. Should we genomically stratify normal ranges to prevent undertreatment of physiological iron deficiency? I don't know if they're all directed at you, Dorine.
Stratified normal ranges.
But Elias, you can come in if you want as well.
Yeah, it's a difficult question.
Well, I'm happy to take it. As I said, unfortunately, I'm not an expert of risk prediction and stratification, but I think the idea seems good. And I'm sure other colleagues are probably already working on this, and it's possibly, in addition to polygenic score, it might be useful to also add non-genetic factor to predict ferritin levels as well.
Yeah, and it should also be said, in addition to that, that even ferritin levels as we use them now are not optimal, because it's not everybody calibrates the assay in the right way. So there's differences between labs. So we all talk about cutoffs, but I mean, we have no good idea of what the cutoff should be in general, even independent from genetically stratified normal ranges. So that's an issue that I think the labs worldwide, the clinical chemistry labs, should solve. That's a different issue from this one. But before we go into detail, maybe we have to solve that first, right? Worldwide.
And Willem, there's a question I was going to ask as well. Can we override this mechanism by giving intravenous iron?
That was my statement with the interleukin 6,
yeah.
that we often use this clinically in people with chronic inflammation. We can override the effect of interleukin 6 by getting iron into the hepatocytes using intravenous iron.
Now, this is a very interesting question. In fact, that's what we do in chronic kidney disease, right? And of course, people in the iron field, makes them very nervous, because it probably has an evolutionary reason why iron is low in case of inflammation. But I must say that from some trials that have been done in chronic kidney disease, for example, the UK pivotal trial shows that it's relatively safe. And you override it because if you give them IV iron, all the iron is going into macrophages and it leaks out, because of course hepcidin is high, will go difficult out of the macrophages, but there's another mechanism that escapes. So ferroportin will be upregulated because of high iron in the macrophages and will leak out. So therefore it works in CKD, and it seems to be in clinical trials relatively safe. But of course we should be very prudent in using it, because it's overriding evolutionary mechanisms, right?
Yes. Now, in terms, as you're there, Sarah, sorry, but in terms of treatment of, you mentioned hepcidin is quite an important peptide hormone. Are there agonists or antagonists of that can be used to modify its activity?
Okay, Yes, of course. I think that all small, but also the big pharma are working on this, either directly, so by directly influencing hepcidin, but also, for example, by influencing hypoxia-inducible factor, because that also indirectly has links to iron metabolism. So I think it's a huge issue. I mean, many companies are working on this, both to treat anemia of chronic disease that is caused by inflammation in let's say prevalent diseases like chronic kidney disease, but also to treat iron overload in thalassemia patients and in hemochromatosis patients. So this is all being addressed. Trials, preclinical studies are going on, but also clinical trials are running throughout the world.
Thank you. And Sarah, did you have a?
Just to say that there's an increasing interest in giving alternate day oral, 1 dose of oral iron and there's quite good clinical trials showing its efficacy, particularly in women with menorrhagia, because it overrides the hepcidin surge that you get when you give 3 times daily, and seems to have very similar clinical effects to 3 times daily oral iron given on the sequential days, and is much better tolerated. So a lot of us clinically are moving to single daily dose of alternate day iron. And I think actually in my practice, that means I less often have to give intravenous iron for iron deficiency, because not only does it work, people actually take it and they tolerate it better.
I think this is we did these studies together with the ETH in Zurich, and I think it's worldwide. I think it's an important remark. I mean, it changes practice. Of course, it can use it very well in, let's say, mild iron deficiency or a mild anemia. If the anemia is, of course, very severe, you might still choose to give it 3 days, 3 times daily, because of course it's all then more iron gets into the body, but relatively lower amounts. Yeah, it's.
Yeah, well, thank you very much. Thank you, Dorine, and thank you, Elias, and thank you for the questioners. I will probably still have my excuse for a steak after I've given my blood donation, but we'll move on now to Nick Gleadall, who will tell us about the results of the BGC Pre-Clinical Study II. To you, Nick. Thanks.
I think Bill is going to go first.
Oh, Bill.
Yeah.
Right. Okay, Bill.
You gave Nick a mild heart attack.
Yes, I know. Keep him on his toes.
All right. So can you see my screen?
Yes, we can, Bill. Yeah.
Great. Awesome. Well, thanks for having me come talk to you guys. I really enjoyed learning more about iron. I didn't know very much about it. So hopefully Nick and I are going to show you guys. We're going to take you through a little bit of the history of how we're making blood group typing possible from the array. I'm going to tell you a little bit about the background and history and what we had done to date, and then Nick's going to show you some of the newer results. So a couple of disclosures so everyone can see. So I thought it'd be important first to just talk what are blood groups. A couple people here might not know, but I assume most do. But quite simply, they're either protein or carbohydrates on the surface of red cells. We organize like-related immunogenic regions into blood groups, often by gene. And these elicit antibody responses and are important for transfusion and also sometimes transplantation, especially the ABO blood group system. So if we're going to tackle blood groups, let's talk a little bit about the numbers. There are over 100 million units of blood collected worldwide each year. There's over 340 antigens, and these are these sort of immunogenic regions on the surface of the red cells. They're organized into 43 blood group systems, controlled by 52 genes, and over 2,000 genetic changes define the expression of these different antigens on the surface of the red cells. So if we talk about current typing methods, the current standard is pretty much unchanged for 100 years, except that we've replaced a lot of patient-derived anti-sera with monoclonal IgM antibodies. But the same basic mechanism applies in terms of testing, mixing red cells with some sort of antibody-based reagent and seeing whether or not they agglutinate, or other types of ways to see whether or not that antibody is reacting with those red cells. And so if you look here on the right, you can see a hypothetical full, pretty full typing of an individual. Many of the antigens that one could type with a plus or a minus sign next to them. And in green are the two that we generally would type, let's say, for this individual. The rest are in red are things that we typically wouldn't know about this particular individual. Now we can do extended serologic typing. It can get very expensive, into the hundreds of dollars. Some of these anti-sera are commercially made, but not at very large quantities, and so they charge quite a bit for them. But you can get some of the additional antigens typed if you do that. There's also a lot of PCR-based assays on the market. Now, a lot of these assays differ in the way in which they read out the detection of the product formed by the PCR, but many of them, or almost all of those assays on the market currently for genotyping, in some way involve a PCR step of some sort. And you can get additional antigens. The major focus of these assays typically are your non-ABO and your non-RHD antigens, but some of them include some small number of RHD and RHC antigens. And they have typically then, because of their formats, require secondary assays to really focus on RH or RHCE more specifically. But you can get more additional changes. But again, the price comes up quite a bit. Our goal was to see how we could try to move this field forward with technology and different assays. And one of our main focuses was, when I started, was really how do we tackle this data? So we created a website called bloodantigens.com. I apologize, anyone who goes to right now, it's down right now. I need to change something on it. I apologize, but it'll be up in the next day or two. One of the things we needed to do was to curate all of the different genetic changes out there in the world, and especially when we started almost a decade ago, this type of information was mostly in PDFs, some on the ISBT website, but often in books and other resources. So our first goal was to look at whole genome sequencing. So we took a trial that we had ongoing with some geneticists. We took those whole genome sequences. We used our bloodantigens.com database, and then we fed it through algorithms we developed called Blood Typer. And then in that case, we were able to then type essentially all genetically defined antigens for the most part in an individual, and you can see now all of that is green. So our goal was, whole genome sequencing is very expensive still, relatively, but if you have that information, it's great to secondarily use it for blood group typing, of course. But as we started meeting Willem and others in this area who were working on arrays, we decided to try to tackle arrays with the same underlying database and typing technology. And we were able to, or at least our initial goal, and I think we've proven this to a large degree, is that one can generally tackle many or not all of the same antigens that you know genetically from an array basis. And the biggest question was, well, how accurate though would it be? And so along with Nick and others in the BGC, we devised a strategy where we took 3 data sets that were existing, some in the UK and some in the Netherlands. And we did an iterative approach where we typed the data set. We looked at what was known clinically about the blood groups in those individuals, improved both the array and the algorithms, the design, all of our methodologies, went on to the next data set and then to the next data set. And so when we first started, the first data set had an overall concordance of 88.9%. And I'm just showing you here the major antigens for which we had serologic or some sort of other genotyping data in the clinical systems for these study individuals that we could do some comparisons for. And you can see for many of the antigens, we are above what I put here as a solid black line at the 95% level. But some were pretty far off, obviously. So we did that iterative changes, and we ran a second data set. And you can see many of those have gone up, and a couple though have come down. But overall, we had an 89.1%. So we did improve a little bit over in the overall direction. And we did that a third time. And you can see now most of all the bars have now gone up, and many of them are now way above 95 percentile. And in fact, overall, the accuracy here was a 99.9% accuracy for these antigens. And this was the largest of the data sets, including over 7,000 individuals, both from England and the Netherlands. So if we look at those remaining discordances that were still there in that 0.9%, the 0.1% of discordances in that data set, we did orthogonal testing, including next-gen sequencing, going back to serology, but see if we could find those samples, if any were archived, or get a follow-up sample. And we binned the discordances into different groups. And I want to just focus on one of them here, and that's the Duffy. You can see here that Duffy was the largest of the discordances in terms of numbers. And that most of those were resolved such that we thought the previous clinical typing was incorrect. And many of those were due to serology calling Duffy weak individuals Duffy negative. And so especially around the Duffy B antigen. And so we feel pretty confident then in these results, in that though there were discordances and some were on the array and some were in the algorithm, we can improve those, and we have. And Nick's going to show you some of those results in a little bit. It's very hard to improve serology much further. We really have, we've been improving serology for years. And so its ability to get improved, much further is sort of limited without major technological leaps. And so we thought this was pretty good results, especially since we were almost, in fact, basically even with serology in terms of the overall numbers of discordances. And so we decided to move forward and really try to understand what was going on here. So if you were to have this information, how would it affect you, right? So it's one thing to be able to accurately get the information, but what would be the effect for a donor system to have this information? And so here's a hypothetical thought experiment, essentially. You have on the top here are your previously typed antigens in the English on the left and the Dutch donor set on the right. And we're only showing here on the x-axis a handful of antigens. These are just the antigens that we had any typing for previously in either of the data sets. And then the rows are the donors in each of those data sets. So these little slivers of lines that you're seeing there are typically maybe even one donor, very faint if you can see them. But as you can see, most people we know mostly ABO and the D antigen, as well as a lot of the other RHC antigens that are commonly typed. And we did enrich these individuals for the people with the most typings, obviously, as well, to get the most bang for the buck. So we mostly picked people who had some extended phenotyping done in a system. You can see on the bottom, though, after running the array on these individuals, now both of those plots are almost essentially solid blue and solid orange, with a smattering of samples where maybe QC didn't pass and the algorithm decided not to make a call on them. But for the most part, this is a wildly different approach in terms of the amount of information one has on their donors. And not only did we have the blood group antigens, the erythroid or HEA antigens, we have platelet antigens on the array, as well as the leukocyte antigens, the HLA antigens. And so this is really a vast increase in the available typing data for these individuals. Prior, we knew about 100,000 individual typings in these individuals, and at the end of running these arrays on these individuals, we knew 1.6 million types in those individuals. And so this was really the impetus for us to say, let's take this to the next step, and we formalized the creation of the Blood Transfusion Genomics Consortium. And here is a graph of the world showing who's currently a member of that organization, as well as for the blood donor centers, blood supply organizations, what the number of donors in their networks are. And so this is really a partnership between both the blood supply organizations, leading academic hospitals, and Thermo Fisher Scientific. And the goal is to develop a simple to use, affordable DNA-based test that could type all those antigens I mentioned earlier. And really importantly, to be able to exploit the rapid decline in the cost of array genotyping technology so that this could be done at clinical scale on donors. And with that, I'll pass over to Nick, and he'll show you the fruits of some of that effort.
Cool. Thanks, Bill. Cool. I'm just going to share my screen. Can everyone see my screen? Yeah?
Yep.
Okay, great, cool. So So yeah, like Bill says, what I'm going to do now is talk about kind of the next steps that we've taken with the array development process and accreditation process. And a quick outline of my talk. I'm first going to introduce the BGC accreditation study for those who don't know what it is. And then I'm going to talk about the donor DNA validation panel that the group, the BGC, the international collaboration has managed to put together. And then I'm going to talk about the human erythrocyte antigen genotyping concordance results from the first phase of the study. So without further ado, so in order to accredit this array and provide some robust validation data, what we've done is we've collected 14,000 donor DNA samples from 7 blood services who are members of the BGC. These samples are then collected in a central lab here at Cambridge University Hospitals, and then they're plated out into triplicate, and then they're sent to one of the 3 BGC genotyping centres. And so these are NHSBT in Colindale, Sanquin in Amsterdam, and the New York Blood Centre. And what we do is once they've been genotyped in triplicate, the data is then sent back to a secure research computing platform that all of the BGC members have access to. And we perform concordance testing results between genotype inferred antigen types and those that were previously on donor record. And what we aim to do with this study is seek regulatory approval for genotyping results to be used for labeling blood units in 2023. So what kind of arrays are we trying to validate with this study? So there are actually 2 arrays that we have. So we have the UK Biobank version 2.2 array, and that's an array that has 800,000 DNA variants on it. So when you type a sample, you type 800,000 positions in their genome, and you can do 96 samples at a time for those. And that's the array that we're using in NHSBT Centre. And what does this have on it? Well, it has the donor and patient typing content that Bill talked about we've been developing over the last few years. And it also has a genome-wide association study grid. So it has variants scattered across the genome so that the data can be used in research studies. That's not suitable for every blood service and every country's regulatory frameworks. So then we also lifted out the donor and patient typing content, and we developed the universal blood donor typing array. And so this is 50,000 DNA variants, but it can do 384 samples. So although you're typing less DNA variants, it's much higher throughput than the other array. And this is the array that we're trialing in the New York Blood Centre and in Sanquin. And like I said before, the content is just donor and patient typing only. So it's only relevant to transfusion. Sorry. So what kind of validation panel have we managed to bring together? So the validation study is going to be done in 2 phases. And the first phase is Pre-Clinical Study II. And so this is the first half of the 14,000 samples that I said we'd collected as a consortium. And so it's composed of about 6,953 samples. That's what we genotyped. And what I've done here on this plot is across the bottom, I've just shown genotype inferred ancestry for those samples, and then the count, how many of them are in each one. And the numbers, the actual exact numbers are labeled above each bar. And so by blood services coming together in the BGC, we're actually able to assemble a much more diverse sample panel than before. The previous publication that we had only 57 individuals in it that were of non-European ancestry in about 7,000 samples. And so you can see that we've really boosted up the number. So 27% of our cohort are now non-European. I think that's a really big thing, because now we can produce extremely robust validation data where we know that the array is going to work in many different populations. Cool. And so this then brings me on to the concordance results. So like I said, the first 6,000 samples have been genotyped. And after QC, we have 5,768 samples. And what I've done here in this plot is I've just pulled out the antigens that as a group, the BGC kind of came together and said, look, these are the diagnostic grade antigens. These are the ones we want to work all the time. And these are the ones that we would likely use for labeling blood units. And so what you can see here is across the bottom, I've just put the antigen. What I've done here is the concordance percentage. So this is the percentage of comparisons that were accurate with the previous typing. So what was already on the blood service donor record. And I've just colored them by different systems, and you can see the key down here. And so in total in this cohort, because we've genotyped everybody in triplicate or a subset also in duplicate, then we were able to do 201,000 comparisons. And if you subset that and get rid of the replicates, and then we talk about unique comparisons, we were able to do 87,979 unique comparisons in there. And again, subsetting the discordances, we had 150 discordances. So overall, from the first preclinical study 2, which we were going to use as a trial of the infrastructure being put into blood services, and was the first real run that blood services had of doing samples in their own laboratories on this technology, we got 99.82% concordance. And so how does that concordance break down, or how do the discordance break down? So like I said before, we had 150 cases where the genotype result didn't agree with what was previously on donor record. And what this plot shows you here, again, I've just put the antigens across the bottom where we observed some discordance. So in the middle of this plot, sorry, there's the antigens. And then the y-axes, they represent the count of discordances. And on the top half of the plot, what I've done is show where the serology or previous typing was positive, but the genotyping array said negative. And in the bottom half of the plot is the opposite. So that's where serology or previous typing said negative, and the genotyping said, no, actually it's positive. And so what you can see is these kind of break down into the same systems that Bill was talking about before, especially given some of them. I think Barbera has already started looking through some of the genotype data. And so for the example, many of these Duffy Bs here, that serology was negative, but genotype was positive, actually by genotype are FYX or Duffy B weak. So we think in that case, what we're going to do is sort of investigate whether the serology was incorrect by confirming the genotypes. And again, in the top half of the plot, we need to investigate, was this something in the digital linkage of the data to the sample, or did the array call these SNPs wrong, or did we call them wrong algorithmically? And so what the plan is for these samples is that now they're going to be distributed to some of the BGC members who have clinical assays in place for resolving these discordances. And so we're going to test them by a third method so that we have a three-way agreement and we can start resolving them and fine-tuning what happened in each case. And then, like I said, fine-tuning the array. So just to summarize that very sort of short tour of the early results, I think the main message that I have from this that really can't be understated in how amazing it is that the genomics infrastructure has been implemented within the clinical laboratories of blood services. This happened over time when COVID was around, and it's just a massive achievement to take a technology that's largely been based within Thermo Fisher's sort of large-scale typing laboratories, and then to put that into a clinical laboratory as a service is quite amazing. The first set of 7,000 samples has been genotyped. We have 150 discordant results in just under 90,000 comparisons. It's extremely encouraging. That's the first trial run of this technology by the blood services. And I really think it's a manageable number for us to go through, of discordances for us to go through and start resolving. And so the next steps, like I said before, the discordant samples are now under investigation. We're going to find out exactly why they were discordant, and the remaining 7,000 samples, the second half of the cohort, the DNA sample panel that we collected, is going to be genotyped in triplicate again by the end of 2022. So that's just a sort of teaser. That's just a sort of early results. This slide is growing and growing all the time. It's a massive effort by a massive group of people. So first of all, I'd really like to say thank you to everyone on this call who's a member of the BGC. I think it's a massive achievement that you've brought together one of the most diverse validation panels I've seen and integrated a genomics technology internationally into several blood services. I'd also like to thank the NHR Bioresource for their constant support of this project as well. So thank you, everybody. And I guess we'll take questions now.
Okay, thank you, Bill and Nick, for these nice presentations. There are no questions in the chat, so everyone who likes to ask a question can open their microphone to ask them directly, I think. Okay, there is a question from Sarah, I see. Excellent work. Will have a real impact on our patient cohort. That's not really a question, but a remark.
Thanks, Sarah. Great.
I love questions like that. That's my favorite.
I think I'll just add to what I was saying before that now we're in this phase of doing a full report. So I presented the HCA antigens that we're really, really interested in working extremely robustly, but the full report will become available within the next month or so, where we do all of the antigens where we have data for, including HLA and HPA as well.
What might be interesting to know, I wonder what your comment on this is. If you send the red cells of 6,000 donors to 3 typing labs where serology is used, and you then look how concordant those results are, we all know that you will get discrepancies. Do you have any feeling for the genotyping, which was done in 3 labs, how good the concordance is between those labs? Are the results in Kansas City the same as in Amsterdam?
Yes.
You looked at that more formally, Nick. So I don't.
Yeah. So I did do that. Obviously, I did that comparison as well. So there's many, many more thousands of comparisons possible. So I think we were able to do about just over 500,000 comparisons, I think, actually in total. And I saw about 90 discordances in that. So the first port of call is to compare back to the gold standard for me, so that we understand where the differences between serology and genotype are coming in. And then the next phase of my analysis is then obviously to look at where genotyping is discordant with itself. What we've also done is, rather than just compare the sort of calculated blood typing results for all of the 800,000 and 50,000 SNPs, we obviously have those in triplicate for every genotype. And so what we can do is measure the genotype to genotype concordance between labs. And for the probes that underpin the diagnostic antigens, that's really excellent concordance there. Yeah.
Yeah, and I think what's important to point out is obviously like something that would almost be impossible. It'd be impossible to do it, right? I mean, just typing for a handful of antigens is like essentially very labor intensive. It's often done manually in many of our labs. You know, when you get beyond just a couple of antigens, you can automate some of it, but most of the antigens, if you want to do a lot of them, you have to do manually. And there you bring in the human factor, and I think that's an important part here, is that this is all being done by the computer and the machines. And so you take out that human factor and having to grade, is it a 0 or is it a 1 or is it a 2+, right? For a lot of these weeks, and especially in these unusual behaviors.
Also that it's in triplicate, right? So you don't have 2,
right.
You've got 3 points of reference to see if they agree or not. So yeah.
That's exactly right. Someone put, Willem, this might be a good question for you. Someone put, this is great, thank you. How much cheaper is the 5K array to the 800K one?
Can I mean, I'm happy. So this is Thermo Fisher. Happy to speak with you. I don't know who the handle is from.
I don't know who that is either. Yeah.
Okay.
Okay.
Yeah.
Send me an email. I mean, like to gather. Definitely, I think it's designed for, you know, it's meant to be the lower cost array, right?
Yeah.
So an 800K. But certainly send me an email and we can speak to it.
Yeah, the reason was I was just excited, and I'm already thinking about implementation when we can plan it in and stuff like that, really, as opposed to anything commercial. Thank you.
Yeah.
Selena had a question, no?
Yeah, Selena did, didn't she?
Yeah, she disappeared on us.
I'm here, sorry.
Yeah.
Carol, I've been meaning to ask this, Nick. Is it also providing genotype, for example, if a predicted pheno is a partial little e, is it also telling us if it's a CEAR versus a CEVS?
I'll defer to Bill, but I think yes.
So yeah, I mean, we're obviously the calling us these complex RH alleles is not necessarily like, if like some of them that have these small little structural variations, like one of the ones you mentioned, is definitely below the resolution which structural variants can be called by the array. That said, often you know, you can call these things by SNPs also, right? And but yeah, if it's able to call an allele, the allele is in the output. Whether we'll go for regulatory approval for that, I'm not sure. What we do want to do is go for regulatory approval to call week D1, week D2, week D3, and some of the alleles. But yeah, all the other alleles are in there, and VVS we are calling out. And it will try to call the allele based on the SNPs present. But even with whole genome or next-gen sequencing, sometimes the VVS alleles are not. There's sometimes still ambiguities there, obviously, right? That's the challenge with alleles, is you have ambiguities even in the best of the data sets. But absolutely.
Thank you.
Yeah.
Okay, there's a question from Pete. What are your plans to complete the validation with samples of ethnicities other than Caucasians?
Well, I think Nick showed a lot of that, right? I mean, we already have, what percent you said, Nick, of the current data set is not
27?
27% of the wave 1 were non-European ancestry, yeah.
This is why we've taken this international approach and try to get many countries across the world involved, so that we could crowdsource those samples and their expertise about what they need in their donors. So yeah, if anyone's interested in that, there's a region of the world you think you could help us with, reach out to Wilhelm or Connie and maybe become a member for future study.
There's a question from Dorine.
From Dorine, yeah, and yeah, of course, this session is about iron, so there's an iron question. Could the panel be used for genotyping of the iron genes in order to personalize for donation?
So the small and the large array have some variants in the HFE locus that were associated with hemochromatosis put on there. The larger array obviously has the GWAS grid sprinkled through those genes, so you can impute from them. Yeah.
Yeah, I mean,
I think I need to correct there.
No.
Both arrays have the full polygenic score for all the iron homeostasis markers on there, and the full polygenic score. So around 250 SNPs across the genome for restless-leg syndrome. So we have worked with the Department of Public Health and Elena's colleagues, who very kindly make these data available before publication.
I think Colin had a question.
My question was the same as Dorine's, because sort of bring it back to iron hemostasis, and we were talking about HFE, and she talked about the transferrin receptors and all the other sort of markers involved, and I think Willem's answered probably my question.
Yeah, we have plenty of room to put more markers on. I think the approach is that when we're going to allow other people to find it on the large format of the array type technology, and then once those things are found and vetted, we have plenty of extra room to throw hundreds of more SNPs onto the other format.
Okay, there's another question about the algorithm.
Yes. So I guess once it's implemented, the typing algorithms will be kind of on board the Gene Titan machines. Is probably the best. That's where they will.
Yeah, yes. So currently it's my algorithm. I've written it over the last decade or so, and we're running it up on the compute cluster. The idea is that there'll be a Thermo Fisher version of that algorithm that would come with the assay and the machines and stuff. And so Thermo Fisher would control the updates for that algorithm in conjunction with the experts in the field.
Okay. I don't think there are any more questions. So I'd like to thank the speakers for their input in this session. It was a great session of more than 100 participants. So that's great. And yeah, thank you all for your attention.
Thank you.
Thank you.
Yeah, okay.